My name is Peng Wang. I'm really sorry that I couldn't be there in person, that because I haven't received my travel document, which is quite sad to me. And but anyway, today I would like to talk about electron ptychography. As you know, the ptychography, many of you know this is one of an emerging technique for physical and biological science. And the tycho here, the word of tycho is fold in Greek, and so which means you have information folded, and the method here is we try to unfold the information inside the data. Okay, so as you know, why we like to do ptychography? The original the idea or the aim is because previously the lens has aberration in it, so the resolution is roughly about 0.1 nanometer. But in last decade, we have developed this aberration corrector, which allow the aberration allow the resolution all the way down to 0.05 nanometer in XY direction, and this is all developed by using hardware, and in order to push the third direction in depth resolution, we have been putting on two double aberration corrector in one microscope, a foamless confocal electron microscope mode. And by doing so, we're able to have very small depth focus so that we can optical sectioning sample along the z-axis and form a three-dimensional imaging. So what we have done actually is in terms of hardware development. As you know, nowadays the computing power has increased dramatically. Not only graphic card, but also algorithms such as AI, machine learning, what have already listening in this workshop. So the question is whether we can push the resolution with aberrated lenses or with a normal microscope. Okay, so therefore, rather than we put all the good high quality optical component on a microscope, so why we go back to a very simple setup, which is just with condensed lens and with a highly defocused beam, and then using a two-dimensional detector to acquire diffraction pattern, rather than use a different and using annular detector to acquire data. So, and this mode is forming like a lensless imaging mode, also called coherent diffraction imaging. And that's bring us to my topic, which is iterative ptychographic imaging. Firstly, I will give a short background of iterative ptychographic imaging, and then we will present a few recent works with a two-dimensional, and then lately we're working towards three-dimensional, and then we also extend towards to hollow ptychography, and we will talk about that in detail. Okay, ptychography is one of coherent diffractive imaging method. Conventional CDI is using to use one diffraction to form an image. However, the detector only detects the intensity and then lost the phase. Therefore, we come to this classical phase problem. And the reason we only have one diffraction pattern, so there is no free lunch here. We have to iterate between the real space and the Fourier space, and with a certain constraint. One of constraint in the diffraction domain is the diffraction pattern in the intensity on the diffraction pattern. And in real space, and what we normally use is use a finite size, such as the sample have to be a particle and with support around which maybe can be a vacuum. So you will find out this method is very useful to the nanoparticles, but it's have a difficulty to apply lateral extended objects. Therefore, with that, we like to investigate that using ptychography, iterative ones. Okay, ptychography plus iterative phase retrieval was originally probed by Hoppe. So in ptychography, rather than using a single diffraction pattern, here we acquire multiple diffraction patterns for many overlapping positions of the object and illumination. So it works well with any instant beam, such as X-ray, laser, or light. And in theory, the resolution is limited by the wavelength of radiation that you use. And we have a few different kind of setup. The setup that we always use is this one. It's a shift illumination by shift the large defocused probe on the sample, and then place the detector in the diffraction domain, and then we acquire a diffraction pattern at different position. And you can find out this scanning mode is very compatible to the current STEM mode in our microscope. Therefore, in my talk, I solely use this STEM mode in the experiment. This is a workflow, as you can see. Rather than we form an atomic size of probe, here we use largely defocused probe so that you have a large probe size instant on the sample. Therefore, you can scan along the sample with relatively large step size. And at each probe position, we acquire a two-dimensional DP. And then because we scan this probe along in two-dimensionally, therefore we form this kind of array of diffraction pattern. And we so-called four-dimensional data set. And using this data set, we use computing algorithm to reconstruct the object function. And this is actually a big data set. As you can see, we scan this two-dimensional, and at each position, we have a two-dimensional diffraction pattern. So this is a big 4D data set. And there are many people using different algorithms to extract information. But here we use ptychography. This is a very simple ptychography iteration flowchart. And as you can see, we begin with a gas function and the probe function. We multiply these two functions together and then Fourier transform this product to the Fourier domain and on the diffraction plane. Because we know the intensity of the diffraction pattern, therefore we can replace the modulus of the calculated diffraction pattern and then keep the phase. And then we inverse Fourier transform to this update function. As you can see, this update function update the area of the object illuminated by the probe at this position. And then we come back to the starting point for the next probe position. So, and then we can iterate until to the last proposition. We call this one iteration, we call this one procedure is one iteration, and then we continue iterate until it's converged. Okay, then we saw some examples because the reconstruction gave you a phase, so which is very phase imaging is very sensitive to light atoms, and the normal way to detect a light atom such as oxygen or hydrogen is using, for example, active CS or ABF or through focal series, or currently much more popular in the STEM mode called IDPC or IPAM, differential phase contrast imaging. But all those imaging techniques relies on high quality lenses. So the question is whether ptychography can offer this without using such high quality lens or microscope. And then we use cerium oxide as an example, a model system, because in cerium oxide, the catalyst particle, the oxygen is the number of atomic number of oxygen is 8, and the cerium is 56. So oxygen is very light, and inside the cerium oxide, in cerium lattice, as you predict in the hard disk, you barely see the oxygen column, but in the face, you able to see a shoulder that is they are oxygen column aside nearby the cerium atomic column. In terms of resolution, I think you can see this is 1 angstrom resolution range, and we are able to see spots. So which means you can achieve atomic resolution with high sensitivity and using this phase imaging technique. And if we further to prove this using lanthanum, this is lab 6 crystal, and the boron is 5, lanthanum is 57. As you can see in hard disk, the lattice is really empty. All the bright spots are corresponding to lanthanum column. Even in the simulation, as you can see, without any noise, you still can't see any boron atomic column. But when you're using ptychography phase reconstruction, you start to see the additional column between the lanthanum. Therefore, in this mode, actually, in this phase imaging, it's really sensitive to light atom and allow you to observe light atom in the heavy atoms crystal. Also, the resolution-wise is also achieve 80 picometer in the resolution. And then we think how we can demonstrate this light atom detection. Then we come to this sort of material. We call this is soft-assembled DNA origami. And in this DNA origami, normally people will, this is belong to organic and inorganic hybrid structure, because sometimes you attack this DNA to the gold particles, which very heavy. For example, normally they will form this functional 3-dimensional structure, hexagonal structure. And then, as you can see, this type of material actually consists of 2 distinguished class material. One is inorganic component, which are metal, oxide particle, etc. And also it contains another class material, organic component. Sometimes it's a ligand, biomolecule, or pharmaceutical substance. And this set of material, class of material, has very important applications in biomedical field. For example, bioimaging and the biosensors, so on. So how to image those ligands, with this heavy atoms particle background. So I think this is quite tricky. In conventional TEM, as you can see, most of people, if the DNA is not stained, so by using conventional TEM imaging, you only see those gold atoms. However, DNA is really, the contrast is really low, at the back, in the background of this carbon support. People always do is using this negative stain, but as you know, when you do negative stain, sometimes you alter the structure, which you cover the real the structure of the biomaterials, and that's why therefore it's challenging to simultaneously image organic and inorganic component in this type of hybrid nanostructure. Certainly, you can't do 2-dimensional. It will be very hard to this in 3-dimensional, because it's difficult to extract this biomaterial component out from the background. Therefore, current three-dimensional imaging of those materials only using, only show those particles or inorganic component in the three-dimensional reconstructions. Okay, so and then that's why we would like to try using ptychography method. So firstly, as you can see, this is the TM mode, and we to quantify this contrast, we sort of using this normalization method. We use background as a 0, and we use the highest contrast density of the gold particle as 1. So we can normalize in intensity overall the image. And as you can see, the contrast or the intensity of the background, which is sitting at 0, and the DNA is in magenta, the separation of these two peaks is too small, which is 0.02. It's very hard to place a threshold between them to separate intensity. Then we try this phase contrast using 300 kV. And as you can see, we get this better. And as you can, we slightly can separate these two peaks, but not entirely. And then we will think whether we can use much lower accelerating voltage, for example, 60 kV. So at 60 kV, we did actually boost contrast both in TEM and then ptychography as well. As if you compare the different settings, at 60 kV, the histogram shows we increase the separations to up to 0.7, but in ptychography at 60 kV, as you can see, we already almost separate the two peaks, and therefore we can place threshold and to extract the organic part from the substrate or from the background, and that's what we did. So in this type of material, we take a 4D data set at each angle, and then we perform ptychography reconstruction for each angle, and we have a two-dimensional data set, a two-dimensional face for that angle as a projection, and then we tilt along different angles in 2 axes, and then we can reconstruct each axis with different angle in terms of face image. And we use current state-of-art tomography software and GenFair developed by John Mell group in UCLA, and we able to reconstruct this 3-dimensional image of DNA origami with the gold particle at the corner. I think it's really good to see that we reconstruct the gold particle together with this DNA component together, especially around. I have to pointing out to you is we're able to see the contrast or DNA component around heavy atom gold particles. So this method, actually, this ptychography method really gave us a high contrast 3D imaging capability, and which can simultaneously image both organic part of and also inorganic part of the some of this hybrid nanostructure materials. Okay. So I think a little bit switch the gear since we talk about 3-dimensional imaging. Actually, ptychography is we have also for ptychography, tomography is not only method we can do ptychography, and there is a unique way for ptychography to extend to 3D imaging, which is we call optical sectioning. Traditional optical sectioning in STEM mode, for STEM mode or confocal mode in the light optics, is by forming a very small point of function, which is a depth of a very small probe with a small depth of focus. And then we scan this sample, a thick sample, in two-dimensional at different depths of focus. So we form this through focal theories of the image, and then we stack them together, form a three-dimensional reconstruction here. But the difference here is in ptychography, we do it computationally, okay? Especially when sample get thicker, we can't use a 2D function to represent the object function. Therefore, we can use this multi-slice approach, which developed by Andy Maiden from Sheffield University. They have demonstrated this method in optical setup. And in this method, we use multiple slices to represent the object function in three-dimensional. Okay, and then electron gonna propagate through this multi-slice sample and then reach to the diffraction pattern. So in this method, and we in terms of electron microscopy field, we demonstrate this method using this two nanotube, this nanotube, one on top of the other, and then without tilting. As you can see, we form this highly defocused probe on the on the nanotube, and then we scan it along in two-dimensional and form this two-dimensional data, this typical diffraction pattern, okay, that we have. Okay, so in algorithm, we computationally generate a sample with number of slices, and then electron propagate through this multiple slice and the Fourier transform to the diffraction plane, and then inverse Fourier transform back, and then back propagate to out of the object. We call it inverse multi-slice approach. Okay, then in this very experiment, we use 6 slices to represent this whole this three-dimensional nanostructure. As you can see in first 3 slices A, B, C, we can see the nanotube oriented in one direction, and then in D, E, F, the second nanotube going the other direction. So which means using this method, we able to separate the nanotubes at different height along the optical axis. The one thing I would like to point out is if you look this 3 image carefully, okay, you can pick up actually the image B and the E actually focus on the nanotube perfectly at different depths. If I zoom in this 2D phase, you can see atomic resolution, okay, in this slice. And as you can see, those lattices actually represent the nanotube walls. And you're also able to see some fine features in the compartment. And this is really a bamboo-like nanotube, actually. And all the features are quite real, as you can see. It's not artifacts, real, and you can see all the the tube or the compartment, the features inside the nanotubes. So it's really quite cool, actually. Using this method, you didn't really focus on any nanotube at all before you start to acquire the data. But within this 3D computational process, and you're able to refocus the sample again at a certain depth. I think this is a very powerful technique. Then we would like to know what's the depth resolution, when we use this algorithm or this in this setup. Then we use we put in more slice into the iteration, and then by looking at Fourier transform at different slice at different depths, and you can see the intensity goes up and then come down. So we can calculate the depth resolution is roughly about 10 nanometer related to this spatial frequency with lattice spacing of 0.02 lattice, which is about 203 angstrom spatial frequency. So the resolution is not that great compared the current setup. It's not that great at the moment compared to a traditional three-dimensional tomography, but it allows you to access three-dimensional information without tilt your specimen or without tilting your holder. Sometimes it's very good for some in situ experiment. But one thing I would like to emphasize here is not just the three-dimensional, but this so-called post-acquisition focusing capability. Because at the beginning of this experiment, we don't have to know how much or what the focus we have on the sample. We just come in and look at the sample at highly defocused condition, and then we just do the experiment. And then we refocus the object at different height using during the post-acquisition processing. We call it post-acquisition focusing. The advantage of doing so is you save a lot of dose budget, when you working on soft material or beam sensitive materials, because a lot of beam sensitive materials, when you image them, when you during the focusing procedure or stage of focusing, and the sample get damaged already. By using this post-focusing, you don't have to focus your sample, so you save a lot of dose budget. So ideally, this is a ptychography method really suitable for beam sensitive method. So that brings us to the dose sensitive materials, because there are a bunch of sensitive materials have really important applications, such as two-dimensional materials and oxide materials or organic, inorganic parasite structures such as solar cells, and also solid state lithium battery. By the time you increase the dose up to 10 to 5 electron volts per square, it starts to damage quickly. The least but not last, which is biological materials. When you shine the sample in, and then you will see a lot of bubbles forming by the electron beams on the biological materials. Then that's why we like to access or examine how dose efficiency or how low dose we can do with a ptychography method. And the way we compare is traditional STEM imaging. As you can see, when we reduce the dose down to 2 to 10 to 3 electron per square, when you're using hard diff, the signal is really, really low. But when you're using ptychography, you can resolve really good contrast of this molybdisulfide two-dimensional materials. The reason, and why is that? I think the reason is, one is, as you can see, when you're using annular detector forming an image, you only use a partial or portion of the electrons that interact with the sample. For example, in hard diff, you only collect the high angle scattered electrons. But a lot of electrons after interacting with sample just went through the detector, doesn't give any contribution to the image. However, in the ptychography, you're going to use all the electrons, which means every electron after interaction with the sample will collect it and will contribute to the final image. And if you do this quantitatively, as you can see, if you draw a line profile across this lattice, as you can see here, and the black curve is the signal, and the red one noise. So the amplitude of signal and actually is identical to the noise. So the signal noise ratio is 1:1. But when you look at the ptychography, so the noise is like 30 times of the noise. So the really ptychography gave a kind of advantage in terms of low dose imaging and really high dose efficient. So, and therefore very suitable for low dose imaging of the soft material. And one of the soft material, I think it will be the biological material, for example, proteins or viral particles. And that is the very first experiment we have carried out at a low dose and at cryogenic temperature for biomaterials. And the material we use are a very standard biological punch freezing method. And then we have this we form this vitrified ice, and then within that we have this viral particle. We transfer this into this in situ cryo holder and cap it at natural temperature. As you can see, this geometry, optical geometry, we didn't use a conventional parallel illumination, and then we acquired TEM image for the image. We use this kind of diffraction mode, and then we require multiple diffraction, and then we use the ptychography imaging method, and we try to reconstruct the face of the particle. In conventional TEM, as you can see, that's the dose we use, which is a 35 electron pounds per square. It's very typical TEM dose for biological imaging. And when we use in ptychography method, as you can see, at a similar dose, 22 electron pounds per square, the contrast of this three-dimensional particle actually is very strong. It's much stronger than as you can see in the TEM. In TEM, the contrast showing this three-dimensional structure, 3D, like a ball, like a two-dimensional disc. It sort of lost this three-dimensional feeling or feature of this particle in terms of two-dimensional projection. However, in Tycho, as you can see this, you got this 3D feelings. You will see, okay, this is a three-dimensional this is a projection of three-dimensional object. And the reason why you can see that is we can have very strong low frequency transfer in the flow rate domain compared to the TEM. Okay. So as you can see, we can see additional peak, which is corresponding to the size of this particle. And to understand this contrast much deeper, I think we just let's go back, take a step back to see the contrast mechanism of conventional contrast of TEM. This is stained adenovirus particle, just show you what the particle look like. Okay, so when you use unstained sample, as you can see, as a moderate defocus, which is very close to focal condition, and you can't see any contrast of this particle actually from the and the reason you can easily to understand that from the contrast function, because the defocus is very close to the focus condition. You got really good transfer at high frequency, but you got really weak transfer at low frequency. As you can see, here is 1/10 nanometer spatial frequency transfer is quite low. If you want to see this particle, presumably what we do normally is we cranking the defocus all the way up to, like, for example, minus 3 microns. And that's where you're able to see some shape. You can see the rough shape of this particle, and that's where the circle is. At this condition, as you can see, by using a large defocus, we do enhance the low frequency transfer. As you can see, we push the contrast function to low frequency. but we sacrifice the high frequency transfer here. As you can see, the first crossing is much lower frequency compared to at a low defocus condition, and we have a massive oscillation at high frequencies. So which means we in terms of high frequency information, it will become much more difficult to interpret. And so, but still the information for the particle, the particle is still low. So therefore, if you want to see the shape of the particle, presumably we have to go much further. We have to push this contrast function much further. Now we have to really cranking up this defocus value further in order to see more higher contrast. As you can see, we can increase all the way up to 25 micron. Therefore, we start to see the shape of the particle. And however, the downside is when you crank the defocus up to here, as you can see from the soaring, all the information actually squeezed down to the low frequency, and the first crossover is about 9 nanometer. So which means you really lost a lot of high frequency signals, high spatial frequencies, as you can see here, right? Above this 0 point, it starts oscillation really heavily. But when you see the ptychography, okay, so that's the ptychography phase imaging data. As you can see, it turns out it's very strong. And then if you look at the information transfer, actually it's way old, up to 1 point something nanometer, to the likewise frequency. So the ptychography phase imaging does give you really good contrast. And then why is that? Then we come back to look this ptychography contrast function again. So this is the contrast function of using the calculation method you can see in SI in this paper. Okay, and this contrast function is using is calculated for converging angle of 1 milliradian with 1 milliradian. As you can see, is within when you use 1 milliradian as a converging angle, your contrast function actually is only on the positive axis, which means it does not have this oscillation, or cross the 0 position, which means the phase will not in the image will not flip as a function of the frequency. I think that's one of the important take-home message from this curve. And the second one is by changing the converging angle, the size of converging angle, which normally you do in the stem mode, you can do in the stem mode. You can tune the bandwidth of this contrast function. Okay, from the low frequency transfer to medium and all the way to high frequency transfer. For example, to 0.2 nanometer when you use 10 milliradian as a converging angle, which means you're able to tune this contrast function using different converging angle. Very similarly, you tune the contrast function in the TM, but using defocus. But the difference is, no matter how you change the converging angle, the contrast function always positive or always on one side of the axis. You don't have such oscillations at high frequency. So that's really one of the advantage of this technique. Okay. So, and to prove this experimentally, and we use a different different converging angles, such as 1, 5, and 10, and we use this full range correlation to see what's the highest resolution we can achieve using different converging angle. As you can see, that we able to achieve this we can able to achieve really high resolution when we use higher converging angle. But just to remind you, when we use to 24 milliradian, we're able to push all the way down to a subatomic resolution. I think will foresee the future of this technique. Okay, because we have such good contrast, okay, therefore we able to continuously decrease the dose when we image biological material. So we're able to decrease all the way to the 5.7. We still can observe the structures of this viral particles. And then we use another set of which are heterogeneous type of viral particles. We, for example, this HIV particle, we also can see this particle, the shape of particle at 5.7 electron per angstrom square. This is really insane kind of low dose, I think, for the biological image. So this is the dose, and as you know, for biology sample, we always have to look the most of interesting features, or actually is within the cells, and the cell is very big. It's quite big. So one of the advantage of this technique is large field of view, because rather than we just form an image with one shot, actually we scanning the probe along a large probe along the sample. Therefore, we able to acquire a data as large as possible. Okay, certainly since we already take this multiple instance of particles, we able to directly reconstruct three-dimensional reconstruction of particle using this so-called single particle analysis way. I think in terms of three-dimensional imaging, I have introduced you like a ptychography, a single particle analysis based on ptychography imaging, and also tomography, and also one of very unique technique for ptychography, which is optical sectioning. I think combine all these three techniques, it's very promising when we work towards Cryo-EM for biological imaging, because currently state-of-art imaging method is using this parallel illumination and forming a TM image to do the single particle analysis or tomography. Therefore, I believe there are in the future there will be a mileage for the application of ptychography in such big area. I think it all sounds very good for with this phase imaging, because we demonstrate this high contrast and also low dose, high dose efficiency for beam sensitive materials like biological. But another thing I think we haven't explored is how can we identify this chemical species simultaneously with this phase imaging at atomic resolution, because when we're talking about phase imaging, we're always using this elastically scattered electron or coherent imaging. But there are also a large portion of electrons which interact with the sample incoherently or elastically. So how can we use those electrons? So combined with ptychography, and that's actually the why that is so successful for STEM hard disk use configuration, because you're able to acquire atom by atom structure information at the same time with the use. You're able to get chemical information atom by atom at atomic resolution. So it's really powerful technique. But for ptychography, because we're using this pixelated detector and then acquire a whole two-dimensional diffraction pattern, basically we don't have any signal allowed to go into spectrometer. So the idea is now whether we can dig a hole inside the pixelated detector, so that we can allow some of the electrons go through the detector and reach this use spectrometer. Therefore, we're able to acquire phase imaging and together with U spectrum. But the question is, when we use this hollow type of detector, whether we still can reconstruct the object function with the limited signal from this annular type of 2D detector. Okay. I think the question, the answer actually is yes. Actually, there is enough information allow us to achieve this atomic resolution-based reconstruction, which is compatible to those reconstruct from whole detector. Okay, when you look at this the Fourier transform, you're able to see the resolution. We can achieve a similar resolution, but the contrast slightly lower than the hole detector. And we have look at how large the hole we can dig, for this very purpose. As you can see, beta is a collection, is a hole inside. But as you know, the larger the hole is, the less signal goes into less signal in the tychography, but more signal goes into use spectrometer. So this is really have a trade-off here. And then we do the calculation. As you can see, if you use beta 5 milliradian or 10 milliradian for the convergence angle like 25, so you pretty much get good, decent resolution and contrast. And then we believe, because most of I think the inelastic scattering actually the characteristic scattering angle of the inelastic scattering is very low. So presumably most of signal will contribute to, hopefully can go through this 10 milliradian hole and reach to electron analog spectrometer and give you enough signal to play with. So with this mode, therefore we kind of propose this a future type of STEM system which has multiple channel, and you don't have to use many annular detectors. You're just using this large pixel data detector with a hole in it and with this yield setup. And by using this, you not only can acquire a hard diff data, and also you can use a DPC, which is very popular currently, and then you can reconstruct phase of using ptychography. And on top of that, you can combine with couple with the use and EDX and whatever you like to do, I think, for this type of system. So with this one, I think I would like to summarize. I think hopefully I will give a flavor of ptychography at different research field. For example, 2-dimensional, 3-dimensional, and all the way to this 5D. 5D here is 4D data set plus 1D energy direction dimensional, which is with the use, which is a multiple channel. So I think in terms of computational microscopy, it's really have a lot of opportunities here. I think I will show you many opportunities when you use ptychography, this method. So with that, I would like to thank my collaborators, and especially thank to my group members and in Nanjing University, and also the people collaborate with us in terms of electron microscopy, such as a big knowledge to Angus Kirkland's group from the Rosalind Franklin Institute, and without their support, I think many of this work couldn't be possible. I also would like to thank Pinellas in Oxford Institute, Oxford University, and also Fu Zhang from Science Tech for the contribution. And many structural biology samples are coming from the people like Jingdong Song from Chinese DC, and Pei Chunzhang from EBIC, Diamond, and David Stewart from structural biology of at University of Oxford, and Chun Haifan from Shanghai Jiao Tong University. And also I would like to thank the funding agency, and also some technical support from Thermo Fisher, and also GEL. And with that, I would like to thank you very much, and I would like to take any questions from you. Thank you.